Did you know that when you combined yellow paint with blue paint, you get green paint? All right, today is Norris Teaches Science Biology Edition video starring me, Mr. Norris, and today I present to you incomplete dominance, and we're going to talk about it and solve them in Punnett squares. So by the end of the video, I can explain how incomplete dominance affects the results when doing Punnett squares. By now, you should have watched the genotype and phenotype video, the complete dominance video, and now you're on to incomplete dominance. We're going to change it up a little bit when we do these Punnett squares because something's going to be different. And let's first talk about what incomplete dominance is. Incomplete dominance results in the blending of traits. So if you look here, here's a red flower, here's a white flower, and they have incomplete dominance, so they would have pink flowers. So it only happens when you have that heterozygous genotype or the heterozygous um, trait. That's where it blends, and it's also it's there because it happens when there's weak or less active genes. So sometimes just because it's dominant doesn't mean it's always going to be dominant, and that's what's going to happen here. So let's solve these. Just jump in. It's good to just do some work and understand it by just looking at how it's happened. So it says in snapdragons, the color red and white flowers show incomplete dominance. Red having the capital letter, that means red is dominant. And lowercase r is white, the recessive trait. So if you have a big R, you're going to be red. If you're big R, you're going to be white. Little r is white, and big R, little r is pink. So in our example, we're going to cross a homozygous red with a homozygous white. Since it says homozygous red, we're going to have the red flower. And homozygous white will be the lowercase r. We don't know which one is which for female or male, so I'm just going to place them in the order they came. Big R at the top, little r on the side. So if we cross, we know that this becomes big R, little r. This is big R, little r, big R, little r, big R, little r. Reason why we know that is because the letter on the top comes down, letter on the side comes over, and we always write them the bigger letter before the lowercase letter. So we have to write a genotype ratio. So the first genotype I see is big R, little r. How many boxes have the big R, little r? Well, all 4 boxes. That would mean that it's 100% big R, little r. Phenotype ratios, remember we have red, we have white, and now since we're introducing incomplete dominance, we're going to have a third phenotype. The third phenotype here is pink. In the question, it will tell you what color the incomplete dominant heterozygous trait would be, just so you don't have to make a guess. So if we look here, red would have to be 2 capital letters. Are there any boxes with 2 capital letters? No, so that's 0%. White would be a box with 2 lowercase letters. I see no boxes with that, so that's 0%. And pink would be anything that's heterozygous, and all 4 boxes are heterozygous, so it's 100% pink. And so this is incomplete dominance example 1. Let's use the same information. Let's do a different one. This time, let's cross 2 heterozygous snapdragons. So if they're heterozygous, we would write them big R, little r. So according to our last slide, what color are heterozygous snapdragons? Pink. All right, let's place the genotypes on the Punnett square real quick. Big R, little r, big R, little r, because it said 2 of them. Pause this video real quick. See if you can solve the Punnett square and write your genotype and phenotype ratios. I'll count down to 5 and I'll begin. 5, 4, 3, 2, 1. Let me go. All right, in this first box here, R's come over, so there's 2 capital R's here. In this next box, R come over, R come down, big R, little r. This next one's big R, little r. And this last one is little r, little r. So let's do the genotype ratios. The first one I see is big R, big R. How many boxes have it? 1. That's 25%. Remember, I use percents here. Next one I see is big R, little r. How many boxes are big R, little r? Well, there's 1, there's 2. That's 50%. And the next one is little r, little r. And if I look, I only see 1 box with that. That's 25%. I double check my math. 25 plus 50 plus 25 equals 100%. Should have got them all. The next one, phenotype ratios. There were 3 colors here. We could be red. We could be white. And we could be pink. All right. To be red, remember you have to be, that's the dominant trait. So you have to be capital R, capital R. How many boxes are capital R, capital R? 1. So red is 25%. To be white, according to our last slide, and we talked about it, it was lowercase r, lowercase r. How many boxes have that? 1. That's 25%. Pink, we already know, is the heterozygous because that's kind of what we did up here. So if we look, there's 2 boxes, 1, 2, that have the big R, little r, or the heterozygous genotype. So that would be 50%. And so if we add those up, we do get 100%. So I know I'm done there. I should have got this all right, and I did because I did make this slide. But this is incomplete dominance. As you noticed, there are now 3 phenotypes instead of the 2 from when you just did complete dominance. But I really hope you learned something today. I would go practice a little bit more. And this is Mr. Norris, and he is signing out.